Okay, next topic, lab on a chip. So introduction of lab on a chip. So lab on a chip refers to technology which allow operations which normally require a laboratory synthesis and analysis of chemicals on a very miniaturized scale within a portable or handheld device. Okay, so which will be with the for the surgery which requires majority laboratory synthesis and analysis of chemicals. Okay, which will be needing many more costly equipment, etc., which will be miniaturized. That is, in the small scale, we are going to use in a lab on a chip. Okay, so by using a single chip, you can able to analyze and synthesize your surgery treatment uh, in a chip level. Okay, that is in the small level. Okay, that will be called as a lab on a chip. So a typical lab on a chip device contains micro channel which allow liquid samples to flow inside the chip. So I think you all know what is chip. Chip is nothing but one single IC. Okay, chip is nothing but single IC. So in that IC you will be having micro channels. So in that you will permit the liquid samples to flow, but also integrates measuring, sensing, and actuating components. So in a single small chip you will be holding all the properties. Okay, that means it will be integrating all the measuring device, sensing device, sensor, actuating component. Actuating means any converter like that. Okay, or analyzer, everything will be implemented in a single chip. So what is LOC? That is lab on a chip. So lab on a chip is nothing but a device which going to integrate one or several laboratory function. ஸோ ஒரு லேப்னு எடுத்துட்டா ஃபார் எக்ஸாம்பிள் இப்போது ஒரு டெலிவரி லேப். ஓகே, இல்லை ஒரு ஹார்ட் பேஷண்ட், ஹார்ட் சர்ஜரி லேப். ஸோ அதுக்கேற்றாப்புல எக்யூப்மெண்ட்ஸ் வில் பி ஃபில்டு இன் யோர் லேப். ஓகே, இப்போ ஹார்ட் சர்ஜரினா ஹார்ட் சர்ஜரிக்கு என்னென்ன எக்யூப்மெண்ட் தேவையோ தட் வில் பி ஆக்குபைட் இன் எ லேப். ஸோ இல்லை ஒரு லேப் பெயின், தட் மீன்ஸ் டெலிவரி. ஸோ டெலிவரி கேஸ்னால் அதுக்கேற்றாப்புல எக்யூப்மெண்ட்ஸ் வில் பி used in the laboratory. So separate laboratory will be used in the hospital. Okay, that will be avoided by using the lab on a chip, because here the lab on a chip is a single device which going to integrate one or more laboratory function. Okay, in a single chip itself. So which will be having millimeter to a square centimeter in size. The chip size will be in millimeter to centimeter size. And the lab on chip technology focuses on the development of hybrid devices which going to integrate fluidic and electronic components on the same chip. Hybrid device means what? So full and full it not going to be acted as a electronic device. So ஒரு எலக்ட்ரானிக் டிவைஸாக ஒரு ஐசி யூஸ் பண்ணாமல் எலக்ட்ரானிக் எக்யூப்மெண்ட்டுக்கு மட்டும் இல்லாமல் இட் வில் பி used for fluidic means you may send the liquid through the same channel. Okay, which going to be injected into your body, the patient body. Okay, so the same channel can be occupied for both electronic purposes as well as for injecting the fluid. Okay, that means medicine. Fluid is nothing but medicine in the same device. So that's why it's called as a hybrid device. So basically, lab on chip integrate non-material, microfluidics, nanosensor, microelectric, biochemistry, fluidic, and electronic components onto the same chip. So all these components are available in a single chip itself. Okay, chip material and fabrication technology. So what are the materials used in implementing or designing the chip? Okay, that is chip material and fabrication. Fabrication na in a how you're producing the chip. Okay, production of chip. So lab on chip devices or hybrids okay, that combine glass, silicon, and various polymer like acrylic, polyester, polycarbonate, resist thermoplastic or mold like the polydimethyl siloxane. ஸோ லேப் ஆன் சிப்புங்கிற டிவைஸ் வந்து ஒரு ஹைப்ரிட் டிவைஸ். ஸோ இதில் வந்து ஒரு செமிகண்டக்டர் மெட்டீரியல் மட்டும் யூஸ் பண்ண போகிறதில்ல. ஸோ செமிகண்டக்டர் மெட்டீரியல்னா இட் வில் பி ஆக்டிங் as the எலக்ட்ரானிக் டிவைஸ். பட் அது செமிகண்டக்டர் மட்டும் யூஸ் பண்ண போகிறதில்ல. ஹியர் வி ஆர் கோயிங் டு யூஸ் த கிளாஸ் மெட்டீரியல், செமிகண்டக்டர் மெட்டீரியல் சிலிக்கான், அப்புறம் பாலிமர்ஸ்னா என்ன, பிளாஸ்டிக். ஓகே, so like acrylic, polyester, okay, polydimethyl siloxane. So likewise you are going to be used for producing the lab on chip device. Okay, silicon, glass, or polymer are suitable for making the microfluidic components of the chip. So these materials will be very suitable for developing the microfluidic component. So microfluidic component is nothing but through that micro channel you can send the liquid, okay, or medicine. So that is possible only through the silicon, glass, or polymer material. Okay, then metal like gold, platinum, or titanium are used for the conductive parts. So conducting material gold, platinum, titanium select பண்ண போறீங்க. So silicon dioxide, silicon nitride, and titanium nitride are used for insulation and passivation. Okay, so non-conducting conducting type la you are going to use silicon dioxide, silicon nitride, and titanium nitride liquids. Then fabrication, how you are going to fabricate the lab and chip, okay, device. Um, See this picture. So you can see different types of lab on chip in the figure. So lab on chip fabrication techniques are analogous to those of microelectronics since closely related to microfabrication and integration methodology are shared by both. There are three way of fabrication process, that is deposition method, etching process, and bonding. ஸோ ஒரு மைக்ரோ ஸோ ஒரு லேப் ஆன் சிப் பார்த்தீங்கன்னா இது ஒரு மைக்ரோ எலக்ட்ரானிக் டிவைஸ் தான். Here we are going to have the three types of fabrication type, that is deposition method, etching method, bonding method. இந்த மூணு மெத்தட்லையும் நம்ம ஃபேப்ரிகேட் பண்ணலாம் டிவைஸ். I think you all know what is deposition. Deposition na layer deposit பண்ணி you will be developing the manufacturing the chip. Etching na இப்போ ஒரு லேயர் போடுறோம், ஒரு மெட்டீரியல் போடுறோம் லேயர், அதில் எச் பண்ணி you will be placing another material. Okay, that will be etching process. Bonding means uh, one material one type of material will be added with another type of material to form a bond. Okay, so in this way in three different types of way you will be developing or manufacturing the chip. Okay, then electronic circuitry on lab on chip. See the diagram, electronic circuit of your lab on chip diagram. Measurement, sensor, signal conditioner product for front end, and then digital signal process. So in between that ADC converter, memory device, bus, this is the transporting device, and then DAC, digital to analog converter, actuator. Okay, so actual input will be fed back to the input side, okay, from the DAC. The sensor is followed by an analog front end, which conditions the measuring signal, analog to digital converter, and a digital signal processor or that analyzes the signal. So measurand will measure the reading. Okay, that will be called as measurand. Any measurement device. Okay, sensor is nothing but sensing any temperature sensor or pressure sensor or flow flow data rate flow will be sensed, everything. Okay, so yeah, particular type நம்ம யூஸ் பண்ண போறதுல, so commonly we are going to call it as a sensor. And then signal conditioning front end na, so the measure which measure the data, that signal will be further processed by the DSP processor. Okay, before doing the DSP processing, you are going to convert the analog signal into digital form, and then only you can be able to do the any processing, like processing means what, any filtering, or if you want to convert one form of signal into another form, that means frequency domain இருந்து time domain க்கு மாத்தணும், இல்ல angular domain, anything else. Okay, any process will be done in the digital signal processor. Okay, so that will be saved in the memories. For that purpose only you are going to use the memory device. And then after doing any process on the digital signal which is converted in the ADC, will be given back to the digital to analog converter, so again it will be converted into analog form. Okay, so again in that, if you get any error in the processing, then it will be given back to the measurement device through the feedback network, that is actuator. Okay, so bus is used for communicating with other channels, okay, or other device, or to the human being, anything else. Okay, for transporting purpose, we'll be using the bus. The signals can be electrical or optical, etc. The analyzed data further sent via bus to external computer for post processing, or even visualized or integrated display or external screen. So the analyzed data from the DSP processor will be sent through the bus to the any external computer for any post processing. Okay, future if you want to do any process on the signal will be done in the computer, or it will be sent to the display device through the bus. So advantages of lab on chip is faster analysis and response time due to short diffusion distances, fast heating, high surface to volume ratio, small heat capacity, better process control because of a faster response of this system. So you can do the control on the process because of the faster analysis and response time. Okay, and then compactness of the system due to integration of much functionality and small volume. So compactness of the system is very uh, good. Okay, and then massive parallelization due to compactness, which allows high throughput analysis. So throughput will be very high in the analysis process because of faster analysis and response time. You will be getting the good compact. Okay, that means you will be doing the more functions simultaneously. Okay, lower fabrication cost, so fabrication cost also very low. And then allowing cost effective disposal chip. So cost effective disposal chip na ஒரு chip வந்து replace பண்றது வந்து ரொம்ப easy. If you are having any failures, okay, you can easily replaceable, the chip is easily replaceable because of its low cost, fabricated in mass production. அதே மாதிரி it can be fabricated in the mass production also. So advantages: low fluid volume consumption, faster analysis and response time due to compactness, massive parallelization. Massive parallelization is simultaneously we have can perform many operations or process. And then lower fabrication cost. So manufacturing cost also very low. And then safer platform for chemical, radioactive, biological survey. So you can use the device for all chemical, that means for sending the fluids, medicines, radio signal, biological signal, anything else. Okay, biological survey means if you want to measure the temperature value, pressure value, ECG value, anything else can be used. can be measured using the lab on view device. Okay, that's the safe platform. And disadvantage in பார்த்தீங்கன்னா novel technology and therefore not yet fully developed. ஸோ இது வந்து ஒரு நாவல் டெக்னாலஜி தான். இதை வந்து இன்னும் we didn't uh, uh, entered into the practice. Okay, ஸோ இது ஒரு நாவல் டெக்னாலஜி தான். ஜஸ்ட் தியரிட்டிக்கலி வி ஆர் analyzed, but it is not completely done practically. And then LOC more complex than in conventional lab equipment. So it will have some complex because we are going to use a single chip for doing major operations means it will be more complex. It will be providing more complex one. And then detection principle may not always scale down in a positive way, leading to low signal to noise ratio. So receiving the signal using detector will slow down the positive way. That means you will not receiving the exact signal. It may cause noise ratio. Okay, SNR problem will arise. That means noise signal we may interrupt with your original signal. Okay, while you are receiving in the detector. So that will be the problem in using lab on chip device. So application பார்த்தீங்கன்னா personalized medicine, point of care, diagnostic. So this chip will be easy to carry by the patient. So அதனால என்ன ஆகும், ஈஸியாக ஒரு டாக்டரால் he can able to diagnose the treatment. Okay, treatment for the disease. And then marine sensor diagnosis means he can able to monitor the data which is collected from the patient easily through the lab on chip. Okay, so marine sensor அங்கே யூஸ் பண்ணலாம். மானிட்டர் பொல்யூஷன், மானிட்டர் பேண்டமிக்ஸ். பேண்டமிக்ஸ்னா என்ன, நோய் பரவுது இல்லையா, லைக் கொரோனா டிசீஸ் monitoring also you can use it. Link to medical and patient database. Data you can collect the data and save the data of patient history in the through the lab on chip device. Okay, to the computer system. Usage as terminal tester. So ஒரு terminal tester-ஆவும் யூஸ் பண்ணலாம் இதை. And then military medicine-ஆவும் யூஸ் பண்ணலாம். So lab on chip can be applied in all this field. Then proposed LOC include HIV test, bacteria test, screening test that can detect chromosome mutation of various cancer. So lab on chip மூலியமா you can do all this test. One of the problem is providing the flow through LOC. The flow of any medicine will be little problem in that. And then instead of using external pump, passive flow occurs when different sized drops are used. That means here in this picture the flow of medicine will cause um, different sized drops. That means it will not be it will not have equivalent drops. Okay, equivalent flow irukade. So to avoid that you may use the external pump and passive flow control. Okay, so if your drop curvature is greater than small drop curvature, then the flow will be in this direction. So large drop curvature, when either large drop curvature less than your small drop curvature means the flow will be in the opposite direction. Okay, this is the diagram of passive pumping device. So flowing fluid in the channel is effectuated by adding a drop to the port opposing the large drop. The subsequent increase of pressure due to the small curvature of the added drop provokes its flow towards the large drop until curvature match. So until the curvature gets equivalent, the flow will be from the small curvature to small drop curvature to large drop curvature. Okay. So it can be taken for the time period second to minutes. And then during the storage of channel, as evaporation occurs both at the large and small drop, that means while you are dropping the liquids, okay, that means while you are storing the drops, what will happen, it may enter into evaporation. Your drops may evaporate, begin to evaporate while you are storing the fluid in the channel, okay, which will be lead to decrease in volume, will provoke more decreases in curvature in the small drop. ஓகே, இதுல இருந்து பார்த்தீங்கன்னா இது சின்னதாக இருக்கும், ஏன்னா எவாப்ரேஷன் ஆகுது. ஓகே, ஆன் ஸ்டோரிங் த லிக்விட் இட் causes to the evaporation so that the size of the small drop curvature will become less, okay, less in size, and thus unbalance of pressure in its favor, a flow will be generated from the large to the small drop. ஸோ அதனால என்ன ஆகும், சைஸ் சின்னதாக அப்படியே ஃப்ளோ டைரக்ஷன் மாறிடும். ஓகே, இங்கே பார்த்தீங்கன்னா ஃப்ளோ டைரக்ஷன் இப்படி இருக்கு, இந்த சைடு இருக்கு, இங்கே ஆனால் நம்ம ட்ராப்ஸ் ஊற்றிட்டு இருக்கோம். ஓகே, ஆனால் கொஞ்ச நேரம் பார்த்தீங்கன்னா நம்ம ஸ்டாப் பண்ணுறோம், ட்ராப்ஸை ஸ்டாப் பண்ணிவிட்டு ஸ்டாப் பண்ணுற டைமில் என்ன ஆகும், எவாப்ரேஷன் ப்ராசஸ் நடக்கும். ஏன்னா நம்ம ஸ்டாப் பண்ணிட்டோம், ஊற்றுறத ஸ்டாப் பண்ணிட்டோம், பட் அங்கே லிக்விட் ஸ்டோரேஜ் இருக்கும் இல்லையா, அந்த ஸ்டோரேஜ்னால இட் மே காஸ் டு எவாப்ரேட். ஸோ எவாப்ரேட் ஆகிறப்ப என்ன ஆகும், இங்கே ட்ராப்பும் இல்லை, அப்போ உன்னோட கர்வேச்சர் சைஸ் குறையும், குறைய குறைய என்ன ஆகும், இந்த ஃப்ளோ ஆஃப் லிக்விட் இங்கேருந்து இங்கே ஊற்றுறப்ப இங்கேருந்து போகும், ஃபில் ஆகும். பட் இப்போ என்ன ஆகும், இங்கே சைஸ் ஊற்றலை, ட்ராப் பண்ணலங்கிறப்ப இங்கே ஃபில் ஆனது இப்போ அப்படியே ஆப்போசிட் டைரக்ஷனில் ஃப்ளோ ஆக ஆரம்பிக்கும். தட்ஸ் த திங். நீ ஏன் ஒரு பைப் மாதிரி ஏதோ ஒன்று ஃபில்லப் பண்ணுற மாதிரி ஒரு டியூப் மாதிரி ஏதோ ஒன்று எடுத்துக்குவேன், அதில் வந்து ஒரு 2 கட்டு ஓப்பனிங் இருக்கும், அந்த ஓப்பனிங்கில் ஒரு ஓப்பனிங்கில் வந்து ஊற்றுறது இல்லை, யூ ஆர் நாட் கோயிங் டு ட்ராப் இட், ஸோ யூ ஆர் கோயிங் டு ட்ராப் இன் அனதர் ஹோல். அப்போ ட்ராப் பண்ணுறப்ப என்ன ஆகும், அங்கேருந்து தான் மூமெண்ட் இருக்கும், அந்த லிக்விட் மூமெண்ட், அந்த ஊற்றுற இல்லையா, போர் பண்ணுற இல்லையா, அங்கேருந்து தான் ஃப்ளோ இருக்கும், இங்கே இன்னொரு ஓட்டைக்கு ஃப்ளோ போகும், அங்கேருந்து ஃப்ளோ ஆகாது, அந்த ஊற்றாத ஓட்டையிலேருந்து ஃப்ளோ ஆகாது, சரியா. இதே வந்து ஃப்ளோ ட்ராப் பண்ணுறல அந்த இதை ஸ்டாப் பண்ணிட்டீங்கன்னா எங்கே ஃபில் ஆனுச்சோ அந்த இடத்துல இருந்து ரிட்டர்ன் வரும் ஃப்ளோ. ஓகே, இதை நீ ப்ராக்டிக்கலாகவும் பார்க்கலாம். த சேம் திங் இஸ் ஃபாலோட் ஹியர் இன் த பாசிவ் பம்பிங் டிவைஸ். So a flow will be generated from the large to the small drop, thus ensuring constant wetting of the port. From Berthier, E.J. Varick, H.U. and D.J. Bibi, managing evaporation for more robust microscale assays. So characterization of convection and diffusion for cell biology. So they done some research on this device, passive pumping device.